أعزائي طلاب المرحلة الثالثة كلية الطب جامعة الموصل، السلام عليكم ورحمة الله وبركاته. أنا الدكتور وليد خالد، تدريسي فرع الأحياء المجهرية في كلية الطب جامعة الموصل. And today I will continue the first lecture about the Enterobacteriaceae. Previously I spoke about the introduction to Enterobacteriaceae. I mentioned the cultural characteristics, the general characters of Enterobacteriaceae in general, the biochemical activities, the antigenic structures. From today, I'll start by giving each member of Enterobacteriaceae in details, and I will start with genus Escherichia. The most important member of genus Escherichia that we concern with is Escherichia coli, E. coli. E. coli is the main microorganism that's present as normal flora in the large bowel stool. These are opportunistic microorganisms. In normal immunity, they are present as normal flora in the intestine. But when the immunity decreases or when they go outside the intestine, then they will become pathogenic, because normal flora are microorganisms that are present normally in certain sites within the body. So migration of these microorganisms to other sites of the body may cause infection. They may become pathogenic if they go outside the enteric, most of enteric bacilli. If they go outside the intestine, they may become pathogenic. Example of this, our example today of Enterobacteriaceae, which is E. coli. Now, morphology of genus Escherichia and E. coli. It is gram-negative bacilli as other Enterobacteriaceae. They are motile and some are capsulated with a slim layer. Regarding motility, we say that all Enterobacteriaceae are motile except Klebsiella and Shigella, which are non-motile. First, we will talk about introduction about the Escherichia coli. We'll talk about the cultural characteristics, the biochemical activity, serological characters, and virulence factors of E. coli. Regarding cultural characteristics, this is the E. coli. They are gram-negative bacilli, as I said. This is how they appear under the microscope. Regarding cultural characteristics, E. coli are facultative anaerobic that grow on simple media. So they are non-fastidious microorganisms. Since they grow on simple media like nutrient agar, they are non-fastidious microorganisms. As I said in the previous lecture, they are lactose fermenters. So on lactose-containing media such as McConkey's agar, they will appear as rose-pink colonies, because, as I said, what happened in McConkey's agar, there's lactose and there's pH indicator, which is a neutral red. E. coli will ferment this lactose, producing acid, changing the color of neutral red to rose-pink color. So E. coli will produce rose-pink colonies on McConkey's agar, as you see here. This is about the cultural characteristics. Now, the biochemical activities. They ferment glucose. They ferment glucose, as I said. This is general for all Enterobacteriaceae. All Enterobacteriaceae will ferment glucose. Also, they are lactose fermenters. As I said, they also ferment maltose, mannate, sucrose, and salicine with production of acid and gas. The in-vic phenomena results of E. coli is positive, positive, negative, negative. As I said, what we mean by in-vic phenomena, it is an abbreviation of four useful reactions that are used to detect the Enterobacteriaceae by their metabolic end products. I represent indole test, M methyl red test, V Vocas-Proskauer test, and C citrate utilization test. So for E. coli, they are indole and methyl red positive, Vocas-Proskauer and citrate utilization test negative. This is the INVIC picture, the INVIC profile of E. coli. This is the indole test, red color ring, positive. Methyl red, also red in color, positive. Vocas-Proskauer and citrate utilization test are negative. Negative Vocas-Proskauer, yellow color, and negative citrate utilization test, green color. This is the INVIC phenomena of E. coli. Positive, positive, negative, negative. Now for each microorganisms, there's INVIC profile. For example, for Klebsiella, it will be negative, negative, positive, positive. So you'll understand what does this mean. It means that they are indole methyl red negative, Vocas-Proskauer citrate utilization test positive. This is for Klebsiella. Today for E. coli, it is positive, positive, negative, negative. This is the INVIC phenomena of E. coli. Now, they are urease negative. E. coli are urease negative, and they do not produce H2S. This is the urease test. The negative test will remain yellow in color, while the positive test, like Proteus volcaris, it will change to pink color. Regarding our microorganism today, E. coli, it will be urease negative. Okay? Klebsiella will be urease positive. We will see this later on. Now, serological characters. As we said, the serological characters are important for determination of pathogenic species and for classification and epidemiological studies. We have three types of serological characters. O, somatic antigen, about 150. Second is the capsular antigen, about 90 of BA and L subtypes. And we have also a flagellar antigen, which is the edge antigen, which is used for motility. We have about 50. Now, the importance of serotyping is to detect the pathogenic strains. Enteropathogenic E. coli possesses O antigen with the numerical designation. The serotypes are written as OK edge. Yani, when we said 111.58 before 2, it means that the O is 111, K capsular antigen is 58, and edge flagellar antigen is 2. Surveillance factors of E. coli, it includes capsule, pili, endotoxins, and exotoxins. Capsule, it will resist the phagocytosis. This is the first virulence factors of E. coli, the capsule. The pili, they are known as colonizing factors. They will use to attach the microorganism to the mucosal surface. They will help the microorganism to attach to the mucosal surface, the pili. The endotoxins and exotoxins, we have two types of exotoxins, enterotoxins and hemolysins. And regarding enterotoxins, we have heat labile toxin, L toxin, heat stable toxin of A and B subtypes, and virotoxin. It is also called as Shiga-like toxin. These are the virulence factors that is used by E. coli to infect the human being. Capsule that help the microorganism to resist phagocytosis. Villi help the microorganism to attach to the mucosal surface. Endotoxin and exotoxin, including enterotoxins and hemolysins. These enterotoxins are determined by plasmid code. Now we'll pass to the second part of our lecture today, which is diseases caused by E. coli, and this is very important for us. They use mainly intestinal infections and urinary tract infections. These are the most important two infections caused by E. coli: intestinal infections and urinary tract infections. Also, they may cause pyogenic infections or sepsis. Now, regarding intestinal infections, the E. coli that cause intestinal infections are classified into 5 types based on O and H antigens. As I said, in serotyping, there's a specific O and H antigen for each strain of E. coli. So, in our case, there are 5 types of E. coli that cause intestinal infections. These the 5 types. We can memorize them by this word, pathy. First strain is enteropathogenic E. coli. Second is enteroaggregative E. coli. Enterotoxigenic E. coli, the third type. Fourth type is enterohemorrhagic E. coli, and fifth type is enteroinvasive E. coli. So we can memorize them by this word, enteropathy E. coli, enteropathogenic E. coli, enteroaggregative E. coli, enterotoxigenic E. coli, enterohemorrhagic E. coli, and enteroinvasive E. coli. And now we will discuss the specific characters of each one of these 5 strains of E. coli that cause intestinal infections. First of all is enteropathogenic E. coli. Enteropathogenic E. coli mainly cause infant diarrhea, and now it is rare in developed countries, but still constitute a main contributor to death to the infant mortality in developing countries. What are the virulence factors that help the enteropathogenic E. coli to infect the intestine? The main factor is by what is called as enteropathogenic E. coli adhesion factor. By this factor, they will attach themselves to the microvilli, disrupting them and causing cupping of the cells around the bacteria, so the intestine will lose its function, its normal function of absorption and secretion. And by this, there will be watery diarrhea occur in case of enteropathogenic E. coli. Okay, so this is the virulence factor by EAF, enteropathogenic E. coli adhesion factor. It will attach to microvilli, causing disruption of them, and then it will prevent the normal function of intestine, which is the absorption, leading to diarrhea. This is about the first strain of E. coli that causes intestinal infection, which is enteropathogenic E. coli. This is the attachment of enteropathogenic E. coli. It will attach by the adhesion factor to the microvilli, resulting in loss of their normal picture, normal morphology, and this will lead to loss of function of intestine, which is absorption, so there will be diarrhea. Second strain of E. coli that cause intestinal infection is enterotoxigenic E. coli. From its name, enterotoxigenic E. coli will produce enterotoxins. We have two types of enterotoxins that are produced by enterotoxigenic E. coli, which is heat labile toxin and heat stable toxin of A and B subtypes. Now, heat labile toxin can withstand temperature up to 60 degrees centigrade for 30 minutes, while heat stable toxin can tolerate temperature up to 100 degrees centigrade. Some strains will produce both heat labile and heat stable toxins, while others can produce only one of them. Labile toxin is similar to the cholera toxin, while stable toxin A will stimulate the activity of guanylate cyclase, leading to increased cyclic GMP production and then diarrhea. This is about the first virulence factors of enterotoxigenic E. coli. The second one is what is called as colonizing factor A, and its role is the same as of colonizing factors that are used by the enteropathogenic E. coli. The CFA, colonizing factor A, will attach themselves to the intestine, so they will prevent the normal peristaltic movement of the intestine, so they will not be delivered outside and they will remain attached to the intestine. By this, they will cause the massive watery diarrhea that is characteristic of enterotoxigenic E. coli, and it is sometimes called as traveler's diarrhea. Traveler's diarrhea is associated with enterotoxigenic E. coli. When someone travels to another country, for example, he may suffer from the traveler's diarrhea. So instead of enjoying his travel to this country, he will stand in front of WC room waiting because of his traveler's diarrhea. This is about the enterotoxigenic E. coli. It will cause massive watery diarrhea called as traveler's diarrhea, and the virulence factors I are by enterotoxins first, and second by colonizing factor A. This is the distribution of traveler's diarrhea in the world, and this is the mechanism. They will activate the guanylate cyclase, leading to increased production of cyclic GMP, leading to massive watery diarrhea. Third strain of E. coli that cause intestinal infection is enteroinvasive E. coli. From the name enteroinvasive E. coli, they will cause penetration, perforation of the mucosa, leading to ulcerous inflammatory lesion. Now, what we will expect from this? We will expect that there will be bloody diarrhea, bacterial dysentery, because it will invade and penetrate the intestinal mucosa, leading to the bloody diarrhea. And the virulence factors is by invasion of the intestinal mucosa. This is about the enteroinvasive E. coli, the third strain of E. coli that causes intestinal infections. Now, the fourth strain is enterohemorrhagic E. coli. This is the invasion by enteroinvasive E. coli, invasion of the intestinal mucosa causing bloody diarrhea. Now, enterohemorrhagic E. coli. Enterohemorrhagic E. coli, it will cause hemorrhagic colitis and hemolytic uremic syndrome. As complication, hemolytic uremic syndrome will occur in 5% of patients that infected by enterohemorrhagic E. coli. Hemolytic uremic syndrome characterized by anemia, thrombocytopenia, and acute renal failure. And the serovar most frequently responsible and associated with hemolytic uremic syndrome is O157H7. This is very important. This is important for you, okay? Because it is important to know this number. So when we isolate E. coli with this serovar O157H7 from a patient, we will put in our mind that this patient is susceptible to hemolytic uremic syndrome. Now, what are the virulence factors of enterohemorrhagic E. coli? It includes the plasmid-coated fimbria that help the enterohemorrhagic E. coli to attach themselves to the enterocytes, and they produce cytotoxins called as virotoxins. And these virotoxins are similar to the Shiga toxin, which is produced by Shigella. So sometimes, in order to differentiate between Shigella and E. coli of this strain, we will name this strain as virotoxin producing E. coli. Most commonly, strains of enterohemorrhagic E. coli have been found on O serogroup with 157, 26, 111, 145, and other serotypes. This is about the enterohemorrhagic E. coli. The final strain of E. coli that is associated with intestinal infection is called as enteroadherent E. coli. And we have two types of enteroadherent E. coli. Diffusely adherent E. coli that is associated with urinary tract infection and diarrhea, and enteroaggregative E. coli that cause watery and sometimes hemorrhagic diarrhea in infants and children. What are the virulence factors? It includes two types. First of all, fimbria, and second, production of toxins similar to the stable toxin of enterotoxigenic E. coli. This is the enteroadherent E. coli. They cause watery or hemorrhagic diarrhea, and the virulence factors including fimbria and toxin production similar to the stable toxin of enterotoxigenic E. coli. And this is the way by which the enteroadherent E. coli cause diarrhea. So, in summary, this is the enterotoxigenic E. coli. They will liberate the enterotoxins. Enterohemorrhagic E. coli, enteroaggregative E. coli, and diffusely adherent E. coli, enteropathogenic E. coli. They will make the microvilli lose their normal morphology, and enteroinvasive E. coli, they will invade the intestinal mucosa. This is the end of part 1, and I will continue with other diseases in part 2.